So I'm going to tell you about our research on Hydra regeneration, and I guess most of you are familiar with regeneration by the work on Planaria, but what I've been doing for the last few years is using electric field or different kinds of electric fields to control Hydra regeneration. And my interest is mainly in the physics of the problem, in the physics of morphogenesis, trying to construct a framework to understand the process of morphogenesis in general, but concentrating on in the context of Hydra regeneration. And what I'll try to do is to walk you through some of our research, starting from electric field effect on regeneration, and then what we concentrate more recently is on the role of noise in morphogenesis. So I hope we'll go through the entire story. I try to give you a comprehensive view, more or less, of what we are doing. But there are many side branches and technical things, so I'll try to avoid technicalities, but if there are questions, please ask. I'll be happy to answer. So this is Hydra. I don't know how much you're familiar with. I'll come back to it in a second, but we are interested in morphogenesis in general, which is a pattern formation process. And especially the question that is behind my research is trying to understand the dynamics of the process, the dynamical framework that allows or ensures the robustness of the emergence of a body form in morphogenesis. And of course, whatever I'm going to show you is impossible to do on mice or whatever. So we choose a model system that is much simpler. This is the Hydra. And I think it's fair to say that the physical mechanisms that drive morphogenesis are poorly understood. This is in contrast to a lot of progress in molecular and cellular biology underlying developmental processes. The physics of the process is much less understood, and this is the aim of my research, is try to advance this. The core physics is very general, so it's not only mechanics, as you'll see. There are many other aspects that come to it. And as I said at the beginning, mainly the role of fluctuations and noise in the back that underlie morphogenesis. So morphogenesis is a process that emerges from the interaction of three types of fields or processes. Not just specific, but three types, I would say. These are biochemical processes, signaling and everything else that is biochemical. Mechanical processes, these are essential in order to form the shape of the body and the three-dimensional adhesion, folding, and etc. that lead to the body form of an animal, and electrical processes that I don't need to convince your group that is important. And electrical processes appear, as you know, in every aspect, actually, in biology. So and this is these two, everybody will agree. This one, I'm not sure that it's a common, how much is a common knowledge, but for your group, it's certainly well understood. But the point is that these fields need to evolve simultaneously as coupled field. So mechanics will not work independent of electrical. Biosignaling will not come independent of mechanics. They are all coupled. in a way that they feed each other, and they need to have enough symbiosis and integration to work as a coordinated process towards a viable organism at the end. So Hydra, I don't know if it's familiar to everybody. I can skip, but anyway, Hydra is a multicellular predator. It's a behaving animal, if you want, which is a very simple geometry. It has something like 100,000 cells with more than 10 cell types. It has a nerve system that is distributed. There is no brain, but there are distributed nerve cells that are connected to each other and to the rest of the system. It has tentacles to catch prey, and a side that is as a mouse, and the side that has a foot that can attach to surfaces. It's a single body axis with radial symmetry, where it has a lot of advantages from my side, trying to understand the physics of the problem. It's very flexible to initial and boundary conditions. We can torture Hydra by many different way, and it will survive and regenerate. It allows control of the dynamics. This is something that is unusual. It's not just perturbing the dynamics or changing it. I'll show you that we can really control it. I mean, we can stop it, we can reverse it, we can move it to different points. And this is not very common in these studies of developmental systems. And the dynamics is really robust, and it allows long-term measurements and statistical view, which is essential as I'll go along. Okay, so this is Hydra. It has a very interesting history, which I'm not going to elaborate on. This is Abraham Tremblay. That he didn't discover Hydra. Hydra was discovered by Leeuwenhoek, that invented the microscope. But he was the first one that discovered regeneration, and he published this beautiful book, which I really recommend for every experimentalist to read. So his experiments on Hydra, he was a mathematician, by the way, a Swiss mathematician. This is a real picture that was drawn by his friend. These are two kids of the Duke of Hague. This is in Hague. He was teaching them, and he found Hydra in the ponds around, and he cut them and started to look, and he discovered regeneration. And this is considered as the birth of experimental biology. This is middle 18th century. And the reason it's a birth of experimental biology, because, and this is the essence of these memoirs, 4 memoirs in this book, that he tell people how to do experiments in biology. Before that, people were only naturalists, looking around, etc. He did real experiments, and he discussed what is a control, how to write a lab book, all the details that we, for us is obvious, but this was the beginning. And what is discovered is this regeneration that you're all familiar. In Hydra, there are 3 modes, if you want, of to run regeneration experiment. You can cut the animal into 2 parts and get back the other half that is missing. You can cut a tissue with a minimal size of, say, 1,000 cells or around a few hundred cells. It folds into a hollow spheroid, which I'll come back to it. It's important. It has to close into a spheroid. If it doesn't close to a spheroid, there is no regeneration. And then within a day or 2, you get the entire animal back. Or you can put it in a blender, dissociate the cells, aggregate them together in a centrifuge, and get again a hollow spheroid and the animal again within time scale of days. So I think this mode makes the Hydra the world champion in regeneration, because to take dissociated cells and get the animal back is something really unusual. I mean, all body regeneration is also rare among the animal kingdom, but this is really unusual. In our lab experiments, we use this mode because this takes more effort, and to get many samples, it's hard. And I didn't find any advantage. If you want to understand this process, going from the spheroid to the entire animal, it's much easier to start from tissue fragments of any geometry. You can invent all kinds of geometries. And this is how it looks in the lab. You take a fragment, this is the hollow spheroid, this is time in hours, and you get the entire animal. Probably you saw this before, but this is how it looks like under our microscope. So the scale here is, we start from a few hundred microns, spheroid, and if you take small tissue sample, you get small animal. You take larger, it's really gradual. Okay, so you can decide what size you're interested in. Study things as a function of size also. So the Hydra body is made out of bilayer epithelial tissues, ectoderm and endoderm, and other types of cell in between. I'm not going to discuss this in detail. What is special about this epithelial system is the fact that these are muscles. So these epithelial cells are also muscles, and these are the actin fibers, supracellular actin fibers. There are two types of actin. One that supports the skeleton of a cell itself, but there are also supracellular actin fibers. These ones run along this is in the ectoderm, they run along the animal, along the axis. This is one animal, we just cut it to get a good imaging of every part. So it goes all the way. These are the tentacles. And in the endoderm, in the inside part, you have the orthogonal supracellular fibers. So there are two fiber muscles that are orthogonal to each other and allows all the movement, contraction, expansion, etc. of the animal. So this is the basic. One last thing about Hydra that will become important momentarily is that it's a freshwater animal. And because it's a freshwater animal, it has to balance osmotic pressure gradients, because the osmolarity, the ionic concentrations inside the cells need to be much larger than the environment. The environment is 1 millimolar of ions, and this requires much larger. So it builds a cavity. That's why the hollow spheroid is really essential, because it's a reservoir of ions that support the animal. It works also as an hydrostatic skeleton for the animal. But these are more or less the ratios of osmolarity. So it's very low outside. It's physiological concentration inside, and in the middle osmolarity in the cavity itself. So water can flow in. You can see this is the area of this spheroid during regeneration. Many times it shows this sawtooth trajectory where it inflates because water comes in, and then it releases the pressure by tearing the tissue. And there are two mechanical forces then that determine the shape of the tissue and the animal at the end. This is hydrostatic pressure by this fluid that is incompressible inside the cavity. that is modulated by osmotic pressure gradients that allow water to flow in and out. And the contractile actomyosin internal forces, these are the muscles. And the balance of these two determines the shape of the body form, of the tissue form, and then again, what morphology can be deformed during morphogenesis. Okay, so what I'm going to tell you is four sections. I'll do them they are not balanced, so I'll see how much of interest you'll have in this. I'll first discuss hydroregeneration under an electric field. I'll do it relatively briefly just to give you the taste what we can do as electric field, external electric field, and how we can control regeneration. And then there are three parts that are important. One is discuss calcium and calcium fluctuations in the tissue. And it turns out because one of the important forces are the internal actomyosin fourth generation, this actomyosin needs calcium to operate as usual in every muscle. And some of the effort went to understand the calcium field in the tissue during regeneration and the fluctuations. And I'll tell you about this. And then this is more going into the physics part. I'm not going to drop on you, fancy physics. I'll just try to demonstrate that morphogenesis is a kind of phase transition. It's a dynamical phase transition, not thermodynamic phase transition. And we'll discuss the dynamics and the model that I think represent well the main physical processes that lead to the morphological transition during regeneration. And then the ideas that come from here and the experimental validation of these ideas show that fluctuations are important, fluctuations in morphology. And this brings me to the last part, which is the role of noise in morphogenesis and the phenomena that is called stochastic resonance that is known in physical systems, but now we can demonstrate it on the entire animal in the context of morphogenesis. And these three parts were done in tight collaboration with this guy, Udeh Dagan, who is a physicist from the Hebrew University in Jerusalem. Okay. And please stop me and ask questions if there are things. So the first thing I would like to discuss is hydromorphogenesis or regeneration under external fields, and show you that we can control the regeneration. And this summarize the process. So we cut a fragment from a mature hydra, a small fragment. It falls into a spheroid. And then the tissue is set between a pair of electrodes. We have many samples. Each one, I don't have the setup. I forgot to put the slide. There are two electrodes covering each tissue sample. In an experiment, we have something like 15. And the multiplexer system that we build, so we can put different voltages and different frequencies, AC frequencies, on each pair of electrode, so we can scan parameters and stuff. But what I'm going to show you is even simpler than that. And what we discovered a few years ago is that there is a critical voltage. Actually, it's a critical field, but the distance between electrodes is fixed, so field and voltage is the same in that case. So there is a critical electric field that if you apply it, you can stop regeneration. Actually, you can stop it at different stages, so it remains a spheroid, and then you release it, you release the voltage, drop it back to 0, and it regenerates. I'll show you a movie in a second. And so this is one mode of control. The other mode of control, which is much more surprising, is that you take an animal or regenerated animal, you apply a voltage above critical. It's not the same number, it's not the same critical value, but there is a critical field above which the animal folds back into a spheroid. It takes the tentacles in, it folds everything, and it goes back. And then once you release it, release the voltage, it regenerates again. And so you can do cycles here, back and forth. Let it develop, going back, let it develop, going back. So these are some pictures. Here we stop regeneration at this voltage, then we release it, it starts to regenerate. Here it's an animal, it folds back, and then we release it. You can, I'll run it fast. So here we stop regeneration, we let the voltage drop to 0, it regenerates. We put higher voltage on it, it goes back, release the voltage, and it regenerates. Okay, so you can go back and forth, and doing a lot of experiments, we check that, for example, this animal, after a few times of regeneration going back, can go mature, can for example, feed itself, can get buds out, and etc. So it's really a reversible, non-destructive process that goes back and forth. Okay, and the numbers are important, but it might there is a lot of variability between tissue samples, etc. So there are no exact numbers for critical voltage. But what is clear is that there is a kind of adaptation, if you want, that if you do a round like that, and then you go to a second round, you need to increase the voltage a little bit. The animal becomes a little bit adapt, but not much. I mean, so gradually you have to increase the voltage applied to the system. And then the question is whether it's just on morphology. This is a big question for a big lab. I'm not a big lab. I'm very small. So what we try to do is to look on the most significant, easy to look signature, which is the head organizer in Hydra. Head organizer in Hydra. Also, it's an interesting history, but before this guy, Spearman and his graduate student, Mangold, found and developed the concept of organizer, which is important in many animals, including mammals. Ethel Brown, in the beginning of the 20th century, did experiments on Hydra and found the process of induction. So you take a head, you take a piece of it, you plug it somewhere else, and you get another head growing from the Hydra, so it can induce. This is, and the essence of organizer, it's a polarity. So it's a signaling center for the tip of the head. In Hydra, it's really in the tip, as you see here. And the molecular biology is more or less understood in Hydra, which I'm not going to get into. But for us, what we did, we used a strain that has a marker for this for the operation of this gene, Wnt3, and which you see here. This is just a fluorescence marker over the body to see that we are not losing the signal because we are losing fluorescence. From the movie, it's hard to see, but under a field, the signal goes away and then it comes back. I'll show you the pictures. It's much better. Maybe I don't know, the movie is not great, but anyway, let's see if I can. You see the signal here coming back after dropping the maybe I can find. You see it already. Anyway, this is the measurement of a few samples. So the fluorescence goes down under the field, and then it gradually comes back when you set the field back to 0. And there is a time scale for this process. But this is just a demonstration that there is hope that the electric field, is affecting processes not only in morphology, but also in the signaling system. So this is part of research that we still need to work out, and how deep it goes into, for example, converting differentiated cells back into stem cells, etc. It's a big, big question. So if you can show, for example, that the external electric field can drive back differentiated cells into stem cells, this will be a very significant discovery. We are not there yet, so I don't know if this really happens. This is just summary for the electric field. So there is a cutoff. So you can go all the way down to DC, but this is a terrible regime to work because it changed the pH of the medium, and the electrodes are coated with all kind of contamination. By the way, all the experiments are done in flow, so the medium is replaced all the time. It's not to keep the conditions constant, but using AC field, there is a cutoff at 3 kilohertz, and this cutoffs comes from the RC time constant of the tissue. So above 3 kilohertz, if you can put zillion volts, and the system becomes completely transparent. The field goes through without affecting anything. And this is because of the filtering, the RC filtering of the tissue. So morphogenesis is certainly reversible, at least from the morphology, and so there are some signs that also from the signaling. But it's reversible in a non-destructive way. So the regeneration potential, it's still very much in operation in all these tissues that go back and forth. The record, by the way, is 5 times. I could go 5 times back and forth between fully developed and back to a spheroid. And after that, because there is no energy supply, just in 1 millimolar of water, it's mineral water if you want, then it loses energy and loses some cells, and I couldn't go beyond 5 times. 5 times is a lot because it goes, develop, go back, develops, go back. And these are also comes very long experiments because I want to see that it develops, fully developed. So it's weeks of experiments on the same samples. And one interesting discovery is that when you go backward and forward, you get many times different body plans, body forms. So it's not, it can start to, as if there are alternative trajectories in development, and you fold it back, it starts a different trajectory. For example, it can go, if by accident I get 2 heads, hydra with 2 heads, I fold it back, I get it back with 1 head. if I get a Hadra with 2 tentacles, I fold it better, I get 5 tentacles with a shape that is not exactly. So there are some signs that there are many alternative trajectories to regeneration. It's certainly not just going back and forth along the same trajectory. The calcium, which I'll come back to it momentarily, is highly intensified by the field. But by the way, the reason a few tens of volts, 20 volts, 30 volts can do all this effect is because there is a natural amplifier in the hydra tissue, which is highly nonlinear, right? Because it's a muscle, so all the epithelium is electrically excitable, like any muscle system. And that's the reason I thought from the beginning that using an external electric field that at low level might excite the system into interesting trajectories, because the system itself needs to be triggered. You don't need to penetrate the system and do all kinds of things. By itself, it starts to propagate electrical activity that is not the regular one. And this is manifested also in the intensified calcium activity, which reflects the electrical excitations in the tissue. Again, there is a decay of the head organizer. That's for sure. How far it goes beyond this, I don't know. And clearly the calcium activity is a very important ingredient, very important field that drives morphogenesis in Hydra. Okay, and I think in many other systems as well, but that's for discussion. There are interesting implications for all this gymnastics. Certainly, one needs to consider morphogenesis beyond biochemical patterning. This is not news for you. And it suggests that morphogenesis is a dynamical process that we need to understand that's something I would like to do, is to try to understand it as a dynamical pattern forming system rather than just a collection of molecular processes that lead to the organism. This is again not news for you. Electrical processes play an instructive role to the level that can direct the developmental process. And the question whether developmental processes are fully reversible is a big question. But even if some of them are reversible, it should lead to some modification in our perspective what development means, because usually the conventional picture of, if you open any book on development, it looks as if it's an hierarchical process that crosses thresholds, and that's it. It goes one way. And then, of course, people were surprised to find out that you can do reprogramming of cells, but the question whether you can do reprogramming of animals, it's a big question. And I'm not saying that I can take a mouse and going back to the egg, that I don't think will happen. But whether it's the real picture or the concept behind development is crossing threshold in a hierarchical way is the right picture, I put a question mark on this, because it might be part of the story, it might be completely wrong, I don't know. But that's the issue.
Can I ask a question? Don't mind.
Sure. But you have to speak louder, or I should, yes.
Are you able, is this okay? Is this
Yes.
loud enough?
Yes.
Yeah. Does the alignment of the Hydra with respect to the electrodes matter at all?
No.
Or if you no?
No. that's why I started with this here. I was hoping that I can direct the process, but I couldn't find any effect of the direction of the electrodes. And even more important, we did a lot of work with Kinneret Keren in our department on the actin system, on the supracellular actin fibers. I was hoping that I could affect their orientation or even melt the order in some regions by the electric field. They don't seem to have any kind of polarity that is sensitive to the direction of the field, or even to the, if it's in DC, to the polarity of the field itself.
The reason it got me thinking is alternating electric field induces a magnetic field also, right? And so the two fields are orthogonal, and then you also have the actin fibers that are also orthogonal.
No, magnetic field is very low here. In another set of experiments, I turned the hydra into magnetic hydra. I coated it with magnetic beads, and then using magnetic probes, you can apply local mechanical deformations. But while doing this, I put an electric field on the hydra. I didn't see any effect, but the field probably is too low. I mean, if you go to 10 Tesla magnetic field, I don't know what will happen. But I don't think it's very important. The electric field is very dramatic effect, and is a direct effect. So this is a strain we built that has a calcium probe in it, the same type of hydra, just with a calcium probe in the epithelium, and you can see the. I want to stop it. It's okay. Look, for example, on this. So the blips you see here, the fluorescence, reflect excitation of calcium activity in the tissue. And to my surprise, the calcium activity is very local. It doesn't spread. There are no waves or something that spread. These waves and spreading occurs only in mature animals. But in the developmental system, during regeneration, it's very local with a very finite correlation length. This is very important because I'll try to convince you that calcium fluctuations are the driving force of the patterning. So if it becomes uniform, if it spreads all over the system in a fast way, then it cannot operate as such. So along the way, these are the type of excitations we find in the hydra. And I want to show you what are the characteristics of these fluctuations. And again, the importance of calcium is important in biology from different reasons. But in our case, first of all, calcium is a mediator between many processes. But in our case, it's a mediator between the electrical activity and the mechanics completely depend on calcium. Without calcium, actomyosin will not operate, will not generate force. So the fluctuations, let me show you a little bit the fluctuations and correlations of the calcium field in hydra during regeneration. We cut a piece, it folds into a spheroid. What you see here, look on the left side, this is at 0 voltage. That's something I showed you before. You see this characteristic sawtooth. This is the area of the tissue, the projected area. The green curves are, the green curve is the mean fluorescence in the tissue over the entire tissue. And the blue is the variance, the spatial variance of the calcium. So it reflects how different parts of the tissue behave in terms of the calcium excitations. And what you see spikes or burst of activity of the calcium at 0 voltage, and this goes all the way, if you let it go, it goes all the way to regeneration. But then we put a voltage that halts regeneration, it remains spherical, and you see a complete change in spectrum of the activity of the calcium, or both the area fluctuations and the calcium activity become completely different. These are, by the way, the definitions of the variants. It's all spatial. And then the tissue remains spheroid. But the great thing about this is that we can hold the system at that state, for example, for days and gather statistics and try to understand the fluctuations and then release it and see how it changes when the system starts to regenerate again. And because we can do it a few times, we can really gather statistics on the process. And this brings me to a second control, which you are also familiar in your group, which is the partial blocking of gap junctions. So in our case, we use heptanol as a blocker with the same family as octanol and all this. I found out that heptanol hydra tolerates it very well, so there is no problem. And to my surprise, not maybe it shouldn't be surprise, not only heptanol halts regeneration, the same as the effect as the electric field, and I come back to it. I'll show you why there are really the opposite effects, because electric field excites the calcium in the system, and heptanol decays the calcium. Both of them halt regeneration, so you can, again, hold it for whatever time you want and study the calcium activity, and then you wash it out and you start to get it again and it regenerates. But the most surprising thing that I'll come to it a bit later is that you apply heptanol to the fully regenerated animal and it falls back. Okay? And it's a question, what are the concentration of heptanol to use, etc. That required a lot of trial and error experiments, because you can destroy the tissue if you really block gap junctions. If you block gap junctions completely, you really disintegrate the tissue. They are really required to keep the system. But anyway, what you see here, you lose this order, spikes and everything by heptanol, you wash it out and you get it back. And these are the summary of the statistics. This is the probability density function. So these are normalized distributions under 4 conditions. The red is electric field, under electric field that halts regeneration. Blue is normal condition. Heptanol is in green. And the black is a tissue in a soft gel that exerts mechanical pressure on the tissue. It allows regeneration, but clearly it produces small animals, and there is mechanical pressure, mechanical forces on the tissue itself. The upper curves, this is the mean, this is the variance, spatial variance. This is the curve, so it's standard deviation over mean, so it's normalized fluctuations. And this is the skewness, so this is the third moment that reflects asymmetric. Again, so everything is spatial. And then we take long time traces. We treat them as statistical ensembles and normalize these curves by the ensemble mean of each of these measure and divide by standard deviation. The reason for that is to get a uniform, a standard distribution with 0 mean and unit standard deviation to compare shapes. I want to see if these shapes are really different. So I normalize them. Certainly, under heptanol, when you block gap junctions, you get a much weaker calcium activity, and also the variance goes down. When you excite it, you get higher. But the question whether these shapes are different cannot be compared like that if you don't normalize. So I want to remove the trivial effect of getting weaker or stronger. And when we do this, this is a nice result. It shows that the normalized fluctuations, the standard deviation over mean, when they normalize properly for all tissue samples, for all conditions, you get a universal curve. So the shape distribution is insensitive. So it's the same shape distribution. The same thing occurs for the right side of the skewness, but not the left side. So there is some asymmetry. I don't know if we'll have time to discuss this, but this is significant because what this tells us is that there are some constraints on the calcium fluctuations. In your language, maybe it's a zero-sum game type of thing. So if part of the tissue is excited, then other parts should be inhibited. So there is a zero-sum game, keeping the shape of the distribution the same for all conditions. We are still not fully understand the constraint, but this is something to take into account in modeling this system. Okay, and the last piece about the calcium is the correlation function, which you see here for the 4 conditions. So this is a length. It's normalized by a length scale that I don't have time to discuss why this length scale is interesting, which is 40% of the tissue, because every tissue will have different size. So to compare correlations, you need to normalize the distance by the fraction of the tissue, if you want, but the length fraction of the tissue. What is clear from here is that under voltage, under external electric field, you get the most extended correlations. Other blocking gap junctions, you get least extended. But at any rate, correlations decay at much smaller length scales than the size of the tissue. This reflects again the locality of the calcium excitation during regeneration. This is just log scale to show you to amplify the differences. Okay, so I'm going to skip. So this is the minimal model. It's an excitable system. It requires 2 fields, activator and inhibitor, with a threshold, nonlinear threshold that you see here. You can ignore the inhibitor by putting everything in a time scale. I'm not going to get into details. What I just want to say is at the end, you get something like a Langevin equation with a potential. This is the derivative of the potential. And you can, I don't know if this language is familiar to you, then you convert it to Fokker-Planck, you get the steady state solution, and the steady state solution, you can derive from it the distributions of the mean variance of all the statistics. And it turns out that the potential you need in order to get the right shapes that we get from the experiment is a double well potential. This is not surprising. This is an excitable system. In an excitable system, you either have 1 fixed point or 3 fixed points. 2 are stable and 1 is unstable. This is the meaning of being excitable. You go from one fixed point to another, and so this is the excitation, okay? And so we can figure out what is the potential. I'm not going to, and get the right shapes of the distributions, more or less. I mean, this is not perfect, so we don't know exactly what is the potential, but we can deduce at least parts of it from the experiment itself. So, okay. So now I'm coming to the, I don't know how much time I have. What's the situation with the time?
Keep going. It's fine. Keep going.
Okay, so now I want to go into the main part. So all this was operative, if you want, to something that I consider as really a very important way of trying to understand morphological transitions during morphogenesis. So again, I cannot generalize it. Let's talk about Hydra. That's what. So in Hydra, this is more or less the timeline. What happens is that you take a fragment, it falls into a spheroid, and then for something like around 48 hours, say, the spheroid just fluctuates around the spherical shape without much happening from the morphology point of view. Certainly, we know that from the molecular and cellular biology, there is a lot going on, a lot. We call it the preparatory stage. But from the morphology point of view, not much happens. And then, after this long period, boom, the spheroid becomes a tube in just a few minutes. It becomes a tube, and this develops into. So you want to understand this transition. It's a geometrical transition, not topological transition, because it's the same topology. But the geometry goes from a spheroid, from a sphere-like to a tube, and this is an essential, or even the major morphological transition that allows regeneration. If you prevent this, that's exactly what the electric field and blocking gap junction is doing, then there is no regeneration. It must go through this transition. Let me show you a movie that shows this transition and how sharp it is. So this is again, I just zoom around the transition point. This is on the left is bright field. This is fluorescence of the same tissue. And you see the time scales here. So it fluctuates. And then at some point, you start to see a little bit of fluctuation to a sphere, and then it goes. And this transition is really very, very sharp. And from this point on, it will make tentacles and all this, the rest of the machinery that makes the mature Hydra. But the point is that this transition is really very sharp. You go, but you could see a little bit of fluctuations that I'll come back to him here. This is a fluctuation, it goes back, and then it goes. Okay. Okay, so in order to study this, we define something that we call the shape parameter. For the physicists, this is analogous to an order parameter in phase transitions, in thermodynamic phase transitions. And the shape parameter that is relevant to our case for this morphological change, we define it as 1 minus the area of the tissue, the projected area, divided by square perimeter of it. And this goes to, this is 0 for an ideal sphere, and it goes to 1 for a long infinite cylinder or tube. And what you see here in this graph, this is time, So there is a long trace before that, which is boring. It just does something like that. It fluctuates around the sphere, a spheroid. Then at some point you start to see some fluctuations, and then boom, it goes. But see how sharp it is? This is a very sharp transition between a spheroid and a tube. And this is summarized under 3 conditions. This is normal condition, no field, nothing. This is partial blocking of gap junctions that doesn't stop the regeneration process, but it affects the system. This is under voltage below critical that allows regeneration, but again affects the system. And what you see, for example, the blue curve here is the average over many tissue samples showing around this transition. So we take many traces, we lock on the transition, make it 0 for all the traces, and then average around it. And you see this characteristic shape. The black line, I'll come back to it, it's a theoretical solution for a model, and it gives you the time scale. And the time scale here is in minutes. So this is, for example, 7.6 minutes transition. So 48 hours, you have nothing, just small fluctuation. Then in a few minutes, it goes. This is a very sharp transition. Under blocking gap junctions, it becomes longer. And in fact, we can play with it a little bit and see that the time scales are clearly affected by blocking gap junctions. And under electric field, we don't have much room to play because I don't want to stop the process. I want to go, but still you see that it's a little bit shorter. But all of them have show this more or less sharp transition, which looks like this hyperbolic tangent, which is a result of a computation that I'll show you in a moment. Okay. So this is something that is of great interest because here we have a morphological transition. It's a major morphological transition that is very important for regeneration. And the question what actually is going on becomes an interesting question, which we can translate into a piece of interesting physics, as I'll show you in a second. But again, I'm not going to bombard you with too much jargon or anything. Okay, any question about this? This is clear. This is the thing we ought to understand and connect it to the
One
calcium.
clarifying question, if I could. So when you
Yeah,
say
sure.
when you say partial gap junction blocking, is this a lesser degree of blocking than you used to take the fully grown, developed Hydra and make it revert to the spheroid?
I didn't show you yet that I can revert the fully developed by blocking gap junction. I'll show you in a moment. I just say that we can. Yes,
But that will be
but
more,
the problem is
more blocking.
the problem with all this, which is also problematic in the case of electric field, is that there are no absolute numbers because each tissue sample has its own individuality, history. This is the problem in general in developmental system in biology. So I don't know absolute numbers. So what I do is I know that above a certain level of heptanol, of blocking gap junctions, I can stop the process. What is the critical concentration? It's tissue dependent. I don't know exactly to tell you a number. So what I do, I reduce it enough to get to see that statistically, most of the samples will just go and do regeneration. But you can play with it. I mean, there is a lot of room to try to understand what really blocking gap junction is doing. I don't know, the sensitivity of it, etc. That's I don't know. I know that if I put too much, I can destroy the sample. If I go below destroying the sample, I can stop regeneration. If I go below this, I get regeneration. It doesn't mean that you take the same concentration that stops regeneration in one tissue samples will not allow regeneration in another sample. This is due to tissue individuality that certainly plays important role. Okay, so I'm not insisting on absolute numbers. I don't care about them. What I care is that I see a real effect. And because we have a calcium probe, you can see immediately that heptanol, that blocking gap junctions affect the system. You see it immediately. I mean, it's not something that I have to guess because I have a probe that tells me something happened to the calcium. Okay, does it answer? I don't know.
It does. I'm leaning towards a bigger question that maybe I'll hold off on, but you know, the quick summary of the bigger question is, playing the devil's advocate, I had expected you to answer Santosh and tell him that you saw processes aligning themselves to an electric field. And when you said that you're not seeing that, then my sort of devil's advocate thought
No, see
is,
what? I didn't hear you. So.
oh, so when Santosh asked before whether you saw any alignment of the morphology to
Yes.
the direction or polarity of the electric field, you said no.
Yes.
And so when I heard that, my thought was that maybe I can play the devil's advocate and suggest that what the electric field was doing was not controlling or directing morphogenesis itself. It was just causing enough cellular damage that the hydra
No. So I'll show you.
reverted to the spherical ball and regrew.
No. Certainly, because I can go back and forth, certainly there's no damage because you get a fully developed hydra, so there's no damage. That's one thing. The other thing is that I'll show you
But it can regrow
momentarily.
itself from shreds. So why does the fact that it is regrowing itself mean that there's no damage? It can regrow from a small damaged piece.
No. First of all, the tissue samples we work with are rather small, so there is no room to go much below. I mean, if it destroys, if I can go 5 times, of course, I'm not arguing that there is no cells that are destroyed in the process. Certainly, but that's not the point. The point to make here is that this is normal regeneration. This is normal regeneration. This is under a field. Certainly, or this is under blocking gap junctions. Certainly, there is an effect, but the process itself looks exactly the same. And what I'm going to show you is that if I block gap junction and apply a field, I can recover the system immediately. So because they work in opposite directions, the field, and also I measure. I know that I put a field, I excite the system, the calcium. When I put blocking gap junctions, I decay the calcium excitation. There is a decay in calcium excitations. Then I can play with these two knobs to find a point where they just cancel each other. I don't know. So I'm not claiming that there is no partial damage or what, because but as I'll argue in a moment, is that I think we understand why the field actually stops regeneration and why blocking gap junctions stop regeneration. But you know, I cannot tell you what happens at the level of the cells or the molecular level or all this. That's I don't know. So it's a fair criticism, but one can do what one can do. That's what I can do.
It sounds good, thanks.
Okay. So the model, let me just give you the essence. I'm not going to go into the physics. So in physics, sometimes we call this an action. So if you integrate over the calcium field, the exponent of this function defines a potential. This is a morphological potential. I want to elaborate a little bit on this concept. It's an important concept. So what processes affect the morphology of the system? Forget all the differentiation of cells, all the biology and everything. Just let's concentrate on the shape of the tissue. As a tissue, I'm not at the level of cells or anything, just the tissue itself. So it contains three parts. There is an elastic part, and for reasons that I don't have time to get, it mainly comes from changing the mean curvature of the tissue, which is kappa here, and kappa 0 is the spontaneous curvature. And the reason for that is that the major elastic process is like buckling process. So the tissue, in order to minimize elastic stresses, it goes out of plane, if you want. It buckles. And this comes, this is taken into account here as the mean curvature. All the rest is not important. This is important because it's incompressible fluid inside, so there is a constraint on the volume, which you have to take into account. Then there is a calcium field, and which is marked as phi here, and this is just a fancy way to write this thing that I've shown you here. Okay, and the reason to write it like that is because we want to connect it to the metric that measures the distances on the surface. But the most important part is this part. It's the interaction part. What is the interaction between the calcium field and the morphology? So this is an hypothesis. It's a guess that the minimal interaction is a direct interaction between the field, the calcium field, and the Gaussian curvature, the intrinsic curvature. So again, I don't know the background. There are two invariant curvatures for a surface. It's the mean curvature, and which is depends on the mean between the two principal curvature of the system, and the Gaussian curvature, which is the multiplication of the two principal. And from the topology point of view, what is important is the intrinsic curvature. The intrinsic curvature changes when the system changes topology. This is not so important. What is important is that this is a very significant type of assumption, because first of all, it assumes some symmetry between the calcium field and the morphology. So writing it in this way assumes that the calcium affects the morphology, but it also is affected by the morphology. And it turns out that this is a very important ingredient. So if you want, if the tissue changes curvature, it affects, it can excite the calcium field. And also excitation of the calcium field can change the curvature. And the symbiotic dynamics between the local curvature and the calcium field is what makes stabilize the system. At any rate, you take the three components, you sum them, and you minimize the system. I'm not going to go into the technicalities, and you can compute this potential. So this is a result of computation. It's not of assuming that these are the interactions in the system. And what you get is a morphological potential, which is a double well potential that you see here here on the right, with this minimum on the left. So this coordinate here is the shape parameter, we have to take it square. I'm not going into details, but this is the shape parameter, which in our case, we assume that it's a single coordinate. This is also a severe assumption, because there could be all kind of metastable states that require more dimension in morphology. But suppose the simpler picture is one-dimensional morphological coordinate. This minimum reflects a spheroid or sphere-like morphology, and this minimum reflects an ellipsoid that goes longer and longer towards a tube. And what we discovered, assuming this, there is a coupling constant here that couples the scalar field, the calcium scalar field, with the curvature, is that as this interaction becomes stronger and stronger, the second minimum becomes lower and lower. And naturally, in the Hydra, this interaction parameter naturally becomes stronger and stronger with time. This is what drives regeneration. So it takes the second minimum here down, 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 until it crosses this one, and then boom, the system makes a transition to a tube. Okay? But the important thing to note here is that there is a barrier. So the system cannot simply go over the barrier if there are not enough fluctuations in morphology. It needs noise in order to overcome the barrier. Okay, otherwise it's locked around this minimum. And this is exactly what happens for a very long time in this preparatory stage until it crosses. I'll come back to it in a moment. But just to complete this picture, you take this potential, you write the dynamics to go from this minimum to this minimum, and this is the solution. These are the black lines I showed you before that that fit the experimental result. This is the hyperbolic tangent that shows this transition, this sharp transition. And it's very sharp because once this crosses fast enough, this minimum goes below this minimum fast enough, then the transition happens. Okay, so this is an important concept of the morphological potential, and you can schematically think about it like that. This is the situation where it's a spheroid. This minimum is too high here, and there's no transition. Then there is a bistable, more or less, situation, and then it goes down. So here fluctuations start to be important, and here it makes the transition. Okay, how do you know that it makes any sense? There are predictions from this model that we can measure. So I'm not going to go into details, but we try to estimate the relation between the calcium excitations and the local curvature of the tissue. And the end result, without going into details, is that indeed you find that in flat regions, you have much more excitation than in curved regions. This is a direct consequence of this type of coupling. To minimize this, you'd like to have negative correlations, which we find between the calcium field and the curvature. So the more curved regions are less excited, and the flat regions are very excited. I'll tell you in a second why this leads to a change in morphology. But then there are other predictions. For example, if we decay this field to become too low, then this integral is just an integral on the closed surface of the curvature. And this is known in topology by a theorem that is called Gauss-Bonnet theorem, that it just gives a number, it's just constant. On the other hand, if phi is uniform, and you can take it out of the integral, it's again tells you there is no change in curvature, in morphology of the tissue. And this is exactly what we find here. So you have a animal that is regenerated. It's a form of a long cylinder. You block gap junctions, and the shape parameter, this is a shape parameter that defined before, goes down very sharply when you block gap junctions. So to maintain the cylinder, you need the internal forces, you need the calcium excitations that are strong enough in the system. It's not a passive thing that once it goes, it maintains itself. It needs the internal forces to keep the cylindrical shape. If you shut them down, then you get more or less a spheroid again. Then at this stage, we apply a field again, an electric field, and it opens the system again. So this demonstrates that we can, with two control parameters that are working one against the other, we can find setting points where you can play with the morphology of the system by either exciting the system or blocking it separately. If we could do it locally, then it would be much more fascinating even, because then we can really make Hydra on demand. We can change the morphology like a plastilina, you know, you can change it by applying these effects in a local way. Let me summarize this and then show you the fluctuation. Can I have another 5 or something minutes? Or it's too late?
Yeah, 5 minutes is fine. We're gonna have to break at 1:30, you know, at 1:30, but 5 minutes is fine.
Okay. So let me tell you the model. The model is a childish thing. If we have high excitation at flat regions more than in curved regions, then the reason it goes into a tube, because it's a pressure system, it's like the balloon party story. You operate the internal forces at flat regions, and then the pressure pushes the system into a tube. Like, if you shut down the forces, then the pressure, because it's isotropic, it takes the system back into a spheroid shape. That's the essence of this. Okay, so now in 5 minutes, the fluctuations. This is high resolution, a high resolution trajectory of the order parameter, of the shape parameter, sorry. So 48 hours, nothing, it's just fluctuates around 0. And then close to the transition, it starts to fluctuate. And we are interested in these fluctuations to understand them. So in order to study these fluctuations, so this occurs very fast. We want to slow down this process in order to study the fluctuations. So what we do, so the reason it goes fast is because the other minimum of the potential goes down fast, very fast, crossing the first minimum, and then the system just goes over the barrier. So in this experiment, we use the electric field to slow down the process. This is highly non-trivial because it's not clear that the electric field can affect this morphological potential or in any way, or whatever way it will affect it. And so what happens is that if I put high enough baseline for the electric field, then I can slow down this change in the tilt of the double well potential. And what you start to see for extended periods, fluctuation between sphere tubes, sphere tube, sphere tube, sphere tube, they become longer and longer because we didn't stop the potential. By the field, I can stop the potential completely. I didn't want to stop it here. I just wanted to slow it down, and then you see the fluctuations, and you can start studying them. So the last part I want to show you, you have a high baseline to slow down the potential, and the next step would be to start modulating it around this baseline. So I'm not going to go into this story. We first found that there is a time scale in the system, which is around 90 minutes. It's a natural time scale. So we modulate the external electric field as a sinus over 90-minute period around the high baseline that slows down the potential. The calcium field that you see down here is fully synchronized with the field. This is the autocorrelation of the calcium field. It's synchronized perfectly with the field. And this is what the morphology is doing. So in orange, you see calcium burst, and in black, the shape parameter around them. And the prediction from this, from a model that runs this, that if you take a particle in a double well potential and you modulate the potential, then if you don't modulate the potential, then the noise takes the particle to the right or to the left, depending if it's strong enough to cross the barrier, to allow crossing the barrier. But the statistics is just Poisson statistics because you get, it's a random process. If you start to modulate the potential, even if you modulate it, you know, with a very weak effect, you just change you just perturb it in a periodic way, in a very weak amplitude compared to the barrier, then you get a resonance. Then you get a resonance between the random transition driven by the noise and the situation of the potential. So you lock the system to the period, and that's exactly what you see here. So this is, and this allows us also to extract the noise level in the system. And so the response as a function of the noise level is a maximum. And the maximum is where the noise level is more or less around the size of the barrier itself. So this is a situation, you modulate the potential. Everything is around, if you want high DC to slow down the process, you modulate the potential where it's more or less at this bistable state, and you get this curve that allows us to extract the level of noise. And what it shows is that the noise is very significant. This is noise that drives the morphological fluctuations in the system. Let me just summarize in 1 minute. So electric field can induce reversal of morphogenesis. I showed you universal calcium spatial fluctuations. We discussed the morphological potential and stochastic morphological swings. These are swings that go back and forth between a spheroid and the tube. And so noise is important. And what is interesting, I don't have time to discuss this in a deep way, is that the noise level naturally is of the order of the barrier height. If this would not be the case, morphogenesis would be impossible in this under these conditions. And the natural timescale of the calcium response is the same timescale if you want for the system to oscillate around the minimum of one minimum of these double well potentials. So this suggests a new mechanism for morphogenesis, which I don't have time to discuss, but I think I gave you enough hints. And again, the theory is with Oded Agam from the Hebrew University. There is a lot of technical help from these guys. And we work also on mechanics and polarity in collaboration with Kinneret Keren from physics.